Okay, so this session is looking at A2 photosynthesis, and with this, the first thing to start with is the word equation. So if you get stuck, come back to the word equation. So you've got carbon dioxide taken in through stomata plus water, which is taken in through the roots, are used in photosynthesis to produce glucose, C6H12O6, plus oxygen gas, which is produced as a byproduct. So if you're stuck, always come back to the word equation. Okay, first part of this, looking at a little animation. So here, if we look in detail at the structure of the leaf with your upper epidermis, cuticle, palisade, spongy mesophyll layer, etc. If we look specifically at a palisade cell, you can see you have many chloroplasts within the palisade cell. If we look in detail at one of these chloroplasts, then we can see that it has an outer membrane, which you'll see here, outer membrane. It's also got an inner membrane, okay? So it's got an intermembrane space similar to a mitochondriae. Inside, you've got these thylakoids, which are stacked in things called grana or granums. You have little links between the stacks called lamella, and then the sort of cytoplasm, if you like, at the center of the chloroplast is known as the stroma. Okay, so here's just a diagram excuse me, of the stomata or the chloroplast you can see. So you've got your outer membrane, your inner membrane, intermembrane space. These are your stacks of thylakoids. There's your lamella joining them together. You might have ribosomes, starch grains, fat, lipid droplets, that type of thing. And the sort of soup, if you like, at the chloroplast is known as the stroma. Okay, so again, stacks of thylakoids, lipid droplets, starch grains, two membranes, stroma in the center. Electron microscope version of that, you can see there's your stacks of thylakoids, your lamella, linking them together, and the stroma, there you can see is the sort of cytoplasm part. Then we talk about the absorption spectrum. Okay, so this is different wavelengths of light that are absorbed by the pigments, which are found within chloroplasts. Now, up to GCSE, you'd normally talk about chlorophyll. You've got different types of chlorophyll. You've also got carotene, which is shown here. You've xanthophylls as well as another example, which it doesn't show. But there are different pigments which absorb different wavelengths of light. You'll notice that most of the absorption occurs in the blue spectrum, so between sort of 400 and 500 nanometers, or 550 most light is absorbed there. And then there's another absorption peak in the red spectrum, sort of between 650 and 700. Notice that in the green spectrum, there's not very much light absorbed. That's why plants appear green. They reflect that green light. Okay, that's linked to the action spectrum. The action spectrum shows the rate of photosynthesis, as you can see here, at different wavelengths. And these work hand in hand, as you would expect. So where most light is absorbed, that's where the highest rate of photosynthesis is going to be. So that blue spectrum and red spectrum, not very much photosynthesis going on in the green spectrum. Okay, now similar to respiration, chlorophyll has, or photosynthesis has stages which follow on from each other. The first one of these is called light harvesting, which we'll look at in a second, which is linked to the light dependent stage. Light dependent stage, as it suggests, means it requires light. And then the final stage, the third stage, is the light independent stage, which, as that suggests, doesn't require light. Okay, there's the word equation just to come back to again. So if we look at light harvesting first, this happens on the thylakoid membrane. Okay, so the membrane of those thylakoids. So within the membrane, you have what are called photosystems. Okay, and these photosystems are often referred to as these funnel type systems, which have what are called accessory pigments, or different types of pigments which absorb light. They pass that energy on by resonance transfer, so they get excited slightly, that they absorb the energy, causes them to move around or vibrate, and then they pass that energy on by resonance transfer. So molecules, other pigments close to it, it'll cause them to vibrate as well, a bit like a domino effect. and that energy gets passed down the funnel to our reaction center, which is always a chlorophyll A molecule, which either is better at absorbing light at a wavelength of P700 or P680. So two different types of photosystems. Okay, a few diagrams here. There's just showing that pass on of energy down through those accessory pigments down to your chlorophyll A or reaction center primary pigment, and that resonance transfer of energy. Now, if we talk about the facts, basic facts of photosynthesis, it's converting light energy from the sun into chemical stored energy, if you like, in terms of organic molecules that are made. The energy gets stored within those organic molecules, so your ATP and glucose. Carbon dioxide is reduced down, so it's absorbed in through the stomata and reduced to produce carbohydrates, and that needs a supply of electrons, hydrogens, and energy, which we'll talk about as part of this light-dependent stage. Okay, so again, this is occurring on the thylakoid membranes. The aim, as you can see, is to produce ATP and electrons, okay, or those electrons are carried by a coenzyme called NADP, and when it picks up those electrons and hydrogens, it becomes reduced NADP or NADPH, okay, and oxygen, thinking about our word equation here, is released as a waste product in the light-dependent stage. Essentially, you've got water, which comes from the roots, is your input. You've got NADP picking up hydrogens to become reduced NADP, and you've got some ATP being synthesized at different stages as well, and then oxygen is released as a waste product. Okay, so in the book they look at this sort of Z scheme as it's talked about. So these are your photosystems, so those sort of funnel systems. When they absorb light, that's passed to the primary pigment. Okay, always chlorophyll A or your reaction center. If enough energy is passed on, it will become very excited and give off an electron. Okay, and that electron is passed through a number of electron carriers within the membrane. At different stages, that produces enough energy to allow ATP to be synthesized, as we'll look at in a moment. If the other photosystem, this other funnel system, absorbs enough light as well, it will give off an electron when it becomes excited. That electron is replaced by this electron coming from photosystem 2 or P680, so it's passed through and replaces that one given off by photosystem 1. That electron is then picked up by NADP, which is that final electron acceptor in photosynthesis. It combines that with hydrogen ions from the breakdown of water to produce reduced NADP or NADPH. Okay, so we talk about water coming in through the roots. It's broken down, and we call that the photolysis of water, into hydrogen ions and oxygen. Okay, or sorry, hydrogens and oxygen. The oxygen is given off as that byproduct and released out through the stomata. The hydrogens split into hydrogen ions or protons, which go towards making reduced NADP, or electrons, which replace these electrons which would have been given off from photosystem 2. Okay, so there is your breakdown of water to produce hydrogen ions and electrons and oxygen. And as I say, those electrons are used to replace electrons given off in the photosystems, and the hydrogen ions are used or picked up by NADP. Okay, so if we look at that, again, using this little animation in a bit of detail. So again, to go through, there's the structure of a leaf, looking at a palisade cell, specifically looking at a chloroplast. And then if we look at what happens on the thylakoid membrane,
here we're looking at a close-up of the thylakoid inner membrane, showing 5 specific points of interest on the electron transport chain. Click on any one of them for more information.
Okay, so if we think of this thylakoid membrane, this is what we're looking at. Sorry, the top here, the thylakoid membrane. Each of these photosystems is one of these funnel light harvesting systems with that primary pigment of either P680 for photosystem 2 or P700 for photosystem 1 at the bottom. Okay, so we take a look at the first bit.
Here you see light from the light harvesting chlorophyll striking the P680 molecule of photosystem 2. P680 loses electrons and becomes oxidized. It captures new electrons or becomes reduced by splitting water, which produces hydrogen plus ions and oxygen.
So we have that energy hitting those that funnel system, enough energy to give off an electron. The electron is then replaced by that photolysis or breakdown of water, which splits into hydrogen ions and electrons, and that oxygen given off as a byproduct.
Shown here are the electrons being passed along a series of electron carriers, which connect photosystem 2 and photosystem 1. Notice that as the electrons move, hydrogen plus ions are dragged into the lumen.
So when the electrons are given off by photosystem 2, they're at high energy levels. There's a number of carriers that they pass through in the thylakoid membrane. That energy is used to pump these hydrogen ions into the lumen or into the center of the thylakoid.
Here you see additional light from the light harvesting chlorophyll striking the P700 molecule of photosystem 1. When this occurs, P700 loses electrons or becomes oxidized.
So here again, light hitting P700 of photosystem 1, that funnel system. Electrons being given off, which are replaced by the electrons which have come from photosystem 2.
The electrons from P700 are passed to the electron acceptor, which then passes them to NADP, which is oxidized, forming NADPH, which is strongly reduced.
Okay, so NADP is the final electron acceptor. It picks up those electrons from photosystem 1 along with some hydrogen ions floating around within the stroma to form reduced NADP.
Represented here is the formation of ATP. Hydrogen ions, which have become concentrated on the inside from the splitting of water during electron transport, diffuse through the ATPase complex, producing ATP.
And then that buildup of hydrogen ions in the lumen of your thylakoid creates concentration gradient. Those hydrogen ions pass through that molecule, that ATP synthase protein. It causes ADP and inorganic phosphate to be bonded together and synthesizes ATP. Okay, again, that's shown here. So here we have our photosystems, or funnel systems. Light hitting or photosystem 2, electrons being given off, which pass through a number of electron carriers, including cytochrome. That electron is replaced by the photolysis or breakdown of water, which produces hydrogen ions and electrons that pass in, oxygen given off as that byproduct. When light hits photosystem 1, again, it gets very excited, gives off an electron. Those electrons are replaced by the ones coming from photosystem 2. Those electrons from photosystem 1 are picked up by NADP, along with some hydrogen ions to produce reduced NADP. That hydrogen ion concentration gradient in the thylakoid lumen allows these hydrogen ions to pass back through into the stroma and synthesizes molecules of ATP at the same time. Okay, so that's the light dependent stage. The light independent stage then, which doesn't require oxygen, but does require the products of the light dependent stage. So it needs energy from ATP, it needs the hydrogens and electrons from reduced NADP, and it also requires carbon dioxide, which is taken in through the stomata. It occurs in the stroma, so in that sort of cytoplasm, if you like, of the chloroplast. Essentially, with the light independent stage, you've got carbon dioxide is the input, and it's fixed along with the breakdown of ATP and the hydrogens and electrons which come from reduced NADP, and then it allows molecules of glucose or organic molecules to be produced. Okay, it's known as the Calvin cycle. So we start with, within the stroma, you have a 5-carbon molecule called RUBP, or ribulose biphosphate. Carbon dioxide is what they call fixed to that with an enzyme called ribisco, the most abundant enzyme on Earth, most important enzyme on Earth as well. That, if you think about that one carbon from carbon dioxide and 5 carbons from RuBP makes a 6-carbon molecule, which is very reactive and splits immediately into 2 3-carbon GP or glycerate phosphate molecules. They go through a number of reactions involving the breakdown of ATP and the dehydrogenation or release of hydrogens of reduced NADP. Those produce triose phosphates, again 2 3-carbon molecules, and then that recycles along with the breakdown of ATP. That's recycled then back to a 5-carbon molecule. So this was 2 3-carbons, that was 6 carbons, and we get 1/6 of a glucose molecule produced for each turn of the Calvin cycle. So in other words, you need 6 turns of this Calvin cycle to produce 1 molecule of glucose. Okay, and that's seen here as well. Your carbon dioxide is fixed to RuBP to produce glycerate phosphate. You get the breakdown of ATP, the oxidation of reduced NADP, so it loses its hydrogens to produce 2 triose phosphates. 1/6 of those is given off to produce organic molecules like glucose, etc. That releases your 5-carbon ribulose biphosphate, which is then converted to ribulose phosphate converted to ribulose biphosphate. So that's the light independent stage. Okay. Some of the graphs that they use, you can see here, this is during light and darkness. Okay, so the concentrations of glycerate phosphate and RuBP are relatively constant when there is enough light. When it starts to get dark, you can see this increase in leveling off of glycerate phosphate, decrease in leveling off of ribulose biphosphate. Okay, so if you think about it, if it's dark, then the light dependent stage doesn't occur, which means we don't get ATP produced and we don't get reduced NADP produced, which means that this part of the reaction here cannot go on. So that's why you get a decrease in RuBP, less of RuBP produced, whereas you get an increase in glycerate phosphate because this part of the reaction, converting RuBP to GP, can still occur. The next graph is then looking at when CO2 is present and when CO2 has been removed. The opposite occurs in this stage, so we get, again, relatively constant. In this one, we get an increase in leveling off of RuBP, a decrease in leveling off of GP. So if we take out the carbon dioxide, then this part of the reaction cannot occur. So the conversion of RuBP to GP doesn't occur, so we get that buildup of ribulose biphosphate. You get this can still occur, this reaction from glycerate phosphate through to RuBP can still occur. So that's why we get that decrease of your glycerate phosphate. Okay, the overview of photosynthesis you can see here. Okay, so within the chloroplast, within the grana, so those stacks of thylakoids, you have the light dependent stage. So light hitting those light harvesting photosystems, oxygen being given off by that breakdown of or photolysis of water. That light dependent stage producing ATP and NADPH, which are then used in the light independent stage in the stroma, carbon dioxide being fixed and it producing those organic compounds or sugars, etc., within the chloroplast. Okay, a couple of experiments that were carried out. So this is Engelmann's experiment to show the different wavelengths of light that were used in photosynthesis. He used a prism to split up white light into blue, green, and red. He also used these special types of cells called spirogyra, which are photosynthetic cells which have this spiral sort of chloroplast. Okay, and he used bacteria that were able to move, which carried out aerobic respiration. Okay, and he found that most bacteria congregated around the chloroplasts where there was blue light. So if you think about these chloroplasts, where there's blue light and also where there's red light, they will photosynthesize, they'll produce oxygen, which then the bacteria can use. So most of the bacteria were found where there was blue light and red light. So that's where most photosynthesis was occurring, whereas green light, with very few bacteria, showing that at those wavelengths of light, not much photosynthesis is occurring. The other one is known as Kelvin's lollipop experiment. And you can see here, this was really to do with the Kelvin cycle at a light independent stage. So he used chlorella, which were a photosynthetic organism or photosynthetic cells. He also used radioactive carbon dioxide, which he added to the chlorella for them to photosynthesize. He controlled these conditions, temperature, pH, etc. And then he stopped, if you like, and took samples of these chlorella at different stages or different times to allow for the different stages of the Kelvin cycle. Okay, and he put them into hot methanol, which killed them. Then he analyzed them to see which substances were present, and that allowed him to identify that glycerate phosphate was produced. Then it was triose phosphate after that, then your ribulose phosphate and your ribulose biphosphate. So he was able to identify those, where the carbon was going, because he used that radioactive carbon. Okay, the last part of this is looking at limiting factors. Okay, so here you can see along the bottom, you've got light intensity. So when we look at this area of our graph, it's the light intensity that is the limiting factor. Low light intensities here, low rate of photosynthesis. As we increase the light intensity, then you can see this first graph starts to level off. So it must be something else limiting apart from light intensity. So in this case, it's more likely carbon dioxide, could be a low temperature as well, but carbon dioxide concentration is very low. If we then increase the carbon dioxide concentration, you can see it follows the same line. So again, before in this area, it's the light intensity that's limiting, whereas when it starts to level off, there's lots of light available here. It's something else. So it's temperature is our limiting factor there. Again, if you increase the temperature and you have again the higher concentration of carbon dioxide, it levels off at a higher level. Okay. In terms of how that works during the day or during different light intensities, when carbon dioxide is given out in this area, you'll have respiration occurring. So that's our respiration. When carbon dioxide is taken in, so this area up the top, that is photosynthesis. Okay, so there's your photosynthesis occurring. And at this level where it's zero, so you've no carbon, a net gain of zero carbon dioxide, so the same amount is given out as taken in, that's where the rate of photosynthesis is the same as respiration, and that's known as the compensation point. So that's the intensity of light where the rate of respiration is the same as the rate of photosynthesis. And if you think about over the course of a day, a 24-hour period, you would have two compensation points. So when the light intensity is very low, so between 12 and about 7 in the morning, light intensity is low, so you have more respiration going on compared to photosynthesis. Whereas after that, you have a higher level of photosynthesis. So there's more carbon dioxide taken in at about 7 in the evening or so, depending on what season it is, about 7 in the evening. That amount of carbon dioxide taken in would decrease as the rate of photosynthesis decreases. And again, your rate of respiration, which pretty much stays the same over the course of the day, it would be higher in this area than the rate of photosynthesis. Hopefully that helps with A2 photosynthesis.